Okay, I think we're on time. Welcome everyone in another episode of Florida Institute Seminar Series. And we're trying to expand our research at UCF through collaborations. And today we've invited Andrew Palmer from Florida Institute of Technology. And then thank you for making the travel today to make this happen. To tell a little bit more about the speaker, Dr. Drew earned his BS in biochemistry from Florida State University, and a PhD in chemistry at Emory University, where his work focused on redox mediated plant signaling strategies. As postdoc, he received NIS fellowship while at the University of Wisconsin Medicine to investigate plant responses to bacterial quorum sensing signals. And since 2011, he started as a professor at Florida Institute of Technology, where his work has transitioned into sustainable space agriculture research. I think we're doing it today.
So thanks for inviting me.
Yours.
It's really nice to be here and share the work that my students are doing with other folks who are in space research. So some of this will probably be stuff you already know in terms of like why we should grow plants in space and do space agriculture, but I always feel it's good to have like our version of that story. We in my lab, we refer to what we do as the red house. So trying to figure out mostly how we're going to grow food on Mars. We've now started to move into lunar, but for a while we really were just solely focused on Mars. That's just how we originally got recruited into the project from Kennedy. Was they came and asked us a bunch of questions about Mars stuff, and I didn't know any answers. Even though I was a big nerd, I was not actually in space biology. But then we started making the migration, and now it's about 75% of what the lab does. So I always like to start with my acknowledgements, just because there's so many people to thank. So this is my lab group as of right now. So we're big. I've got 2 PhD students, 6 master's students, and 15 undergrads. The undergrads are a core group of the research that you're going to see today. None of it would be possible. Actually, one of these guys, this guy here is my first PhD student doing space biology. All the work you're going to see today has been done by undergrads. And so this has really been a real testament to those students. I can't thank all the different people that we work with enough. We have tons of partners at other places. We work with some people at Exolith. We have the Plant the Moon Challenge. We've gotten a lot of friends by being on the scientific advisory board for that. Anka Delgado is a compatriot of mine at Arizona State, and we have a project that I'll talk very briefly about at the end. And so many other people that are just so integral to what we do. And I'll try to pepper in names as they come up. So, why should we go to Mars? Well, there's a bunch of different arguments that are made. We have to worry about overpopulation on the Earth, though I'm not sure Mars is where you would want to solve that problem. The opportunities for resource mining. From a scientific perspective, we're really more interested in the potential for what we can learn about the origins of life, and now possibly more about the persistence of life, right? And arguably, it's what comes after the moon. We get to the moon, we have settlements there. Where do we go to next? Mars seems like a reasonable possibility. So when I first got involved in this, we started talking about the challenges of supplying a colony. Well, I really hadn't thought it through until I was sitting at Kennedy Space Center and they were telling me about the problem. They're like, well, this trip takes about 6 to 8 months. The radiation exposure could have significant consequences on whatever food you're going to bring. Missed shipments in that kind of environment could cost lives. Very easy to imagine a situation where a rocket can't make it back to Mars, you miss by a month or two, and people die on the settlement as a result, right? So space is at a premium on these missions. So how expensive is this process of having to continually resupply a settlement that's so far away, that's such a challenge? What does that look like? So here's some back of the envelope kind of calculations I made a while back. There was a Nature paper in 2015 that estimated that at current costs for upmass, it would be about $17,000 to mail a letter to Mars, and that was 2015 numbers. So That means 5 ounces of arugula would cost you about $50,000. 5 ounces of arugula is about 50 calories. I like arugula, so, but that means 2,000 calories worth of arugula would be about $500,000 a day, or $5 million a day for 10 settlers, right? So that's expensive. And we don't really know what the Martian caloric requirements are going to look like. Really, that's an unknown factor. There's numbers out there from theoretical dietitians, but that's kind of a weird field. And there's a lot we don't know about exercise, food consumption, what the dietary needs are going to be on the planet. And fresh food won't survive the trip anyway, so you won't be able to take that much arugula with you. There's also the rare possibility that you might want to eat something other than arugula while you are stuck on Mars. Sorry, while you have the joy of being on Mars. All right. So as a result, in situ food production is going to be mission critical. We have to provide food security to the colony with everything else that they're dealing with. You cannot have them stressed out about where the next meal is coming from, or if there is a next meal coming anytime soon, right? So we also need to be providing increased variety in the menu options. There's some extra benefits to this process. So one of the first benefits that is frequently touted is life support assistance through CO2 uptake. So plants, of course, do photosynthesis, so they can help decrease CO2 levels. Something that doesn't get talked about as much is that while the atmospheric density on Mars is low, the concentration of CO2 is relatively high. So it would actually be possible to build carbon, which is sort of from an ecological, biological standpoint, sort of the fundamental building block, which is a limiting component to accumulating biomass, is carbon uptake. We could actually grow the colony off of Martian-derived CO2. So within a few generations of plants, everyone will have consumed Mars CO2s, which means all of our settlers will be Martians, literally, because they will have Martian CO2 as part of their biology. Right. So another benefit, and this is one I think that is maybe not as well conceived, is our understanding of how pharmacology works and how pharmaceutical compounds will be preserved over spaceflight is not clear. There's a little bit of evidence out there that suggests that certain drugs don't survive as well. They lose efficacy while in space. So it's not clear if we send things, aspirin or other treatments, to a Martian settlement, that they'll arrive there and still have the same efficacy. So what we can do instead is envision a biomanufacturing molecular farming approach where we can start engineering plants to produce things we need. There's also unexpected things that might occur while we're there, right? So we can have as tested people as much as possible and done tons of psychological screening. It's very possible that someone could develop depression or anxiety while they're in this stressful environment. It'd be really great if we had a plant-based system for producing certain pharmacological compounds, or an algae-based system to do that, where we could provide people needed pharmaceuticals for some temporary period of time, antimicrobials, right? So these are a lot of things, even aspirin. It would be useful to be able to derive that on site from some sort of source, some biological engineered source. All right. A third benefit, psychological benefits and crew morale. So there's been tons of evidence that shows that on the space station, just being around the plants cheers people up. We kind of knew that was going to be the case actually in advance, because if you talk to anybody at any of these isolated stations like McMurdo or anywhere like in the Antarctic, the places that have greenhouses, people just set up shop and hang out in there. They bring a chair, they sit and they read a book because there's green there, right? And there's a fundamental connection to that concept of being near nature, which is probably going to be an even bigger component when you are on a place like Mars, where none of the sky doesn't look the same way. There's no greenery around unless you're in those spaces, right? All right. So how do we develop food security? And so we spend a lot of time talking to some of the engineers at Kennedy about how do we provide food. And one of the constant arguments we get is, well, we're going to use hydroponics. Why? Because hydroponics works. And I say, well, hydroponics works until you lose power. And then we have some other issues to worry about. And really, though, what we're talking about is that true food security, true resiliency is going to depend upon not relying on a single food production strategy. So actually what we're going to need is hydroponics, which are certainly not going to be out on this sort of on the surface greenhouse on Mars. Pretty much our models all, and I don't know anybody who really disagrees, is that most of our growing is going to be done probably underground or in closed chambers. So we're going to be relying on all artificial lighting for growing plants in space, or at least on Mars. Some potential aquaponic strategy, but really we're talking about the difference between hydroponics and what we're going to call for this talk regolith-based agriculture, growing in the surface material that's available, in this case on Mars. We're going to spend most of this talk talking about Mars, not so much the moon. So what we're trading off here is between the NASA beloved term technological readiness level, or TLR, or TRL, sorry, and how resilient is the system. Resilience depends on having a continuum of food options, right, that have different requirements, somewhat different inputs, and somewhat different limitations, right? So we're going to live here today. I'll tell you that our lab is actually working with a couple of other groups, looking at buildings that models of efficiency to actually compare directly side by side, compare hydroponics and regolith. But that project, we really just got our first data set from that a couple of weeks ago. Like literally, I am analyzing it now. So this all started as a project we called Radish, or Research to Advance the Development of Interstellar Horticulture, which started off a few years ago at Florida Tech at a workshop where we were working with Kennedy and decided, yes, we were going to make a play for this and start answering questions. I have to thank this club on campus, the Astrobiological Research and Education Society, Aries. They really gave us the first student workers for this project, and they've continued to be a real nice partnership on campus.
Did you come up with that acronym?
I came up with the acronym, the Radish one. I didn't come up with the Aries one.
That's on point, that Radish one.
Yeah, I have to admit, I sat at the workshop writing things down. I was like, I need a good acronym. I was like, so, all right. So we're going to use Martian regolith for food. Why? Or sorry, for food production. We're not going to eat regolith. So it's a substrate that's already available on Mars in abundance. And our model, in part, is that this is a less expensive process than hydroponics. So if you contemplate, again, the costs of upmass, right now it would probably cost about $150,000 a pound to send equipment to Mars to do hydroponics work. Now, we could talk about ways to 3D print or build materials for that, like pots and structures off of material that's there. That's a very prominent concept in ISRU that I think is really good. So we want to bring those costs as much as possible. This is not about saying hydroponic sucks and regolith is good. So this is about comparing, talking about what we can do with regolith. In our model, you need both systems. All right. The nice thing about incorporating a regolith-based agriculture system is it allows us to incorporate the microorganisms that we know already are involved in agricultural and biological processes here on Earth, in soil, and in relationship to plants. We can develop native carbon, phosphate, and nitrogen cycles, which will actually help us improve nutrient cycling. And rather than trying to redesign the wheel for all of our waste processing and all of our nutrient cycling, we could do what Earth already does and what we've learned, basics of the process, right? And so we can try and build on that and allow it to be a more sustainable system because you have more resiliency in the parts of the biological components. The other reason is that regolith actually contains useful nutrients for plants, which would reduce transport costs. One of the things we don't talk about a lot in hydroponics is you need to bring the nutrient solutions with you. Making a nitrogen fertilizer factory on Mars, which tend to be explosive, is probably not necessarily a great place to do that, right? So where you're going to get those nutrients from is critical, right? So if from a plant biology perspective, we've got a nice amount of iron, not surprising, right? We've got, calcium, we've got magnesium, we've got some phosphate, sulfur, so there's potassium, there's a lot we can work with here if we can get it out. And that's something I'll talk about in a second. The other reason it's good enough for Matt grew stuff in regolith, right? So if Matt Damon can do it, surely we can do it. So, if any of you have watched The Martian, I always think of it, it's a very inspiring movie. all the food had to have gotten irradiated on the way there, they would never have grown. So it's a sad thing, that would have died. But, so, of course, and many of y'all probably know this, the problem is that we don't have any Martian regolith. I've actually had in several talks had to explain to people that we have not brought Martian regolith back, so we don't have any to play with yet, right? If you think about how much we have in terms of lunar regolith, right? The first time plants were grown in lunar regolith was less than a year ago, right? Annalisa Paul's lab at UF, that's the first time, right? That's impressive that, that's the most that we've been able to do. So think about, we're going to come back from Mars with far less when we do have a return mission. So instead, like y'all, we rely on Martian regolith simulants, right? And these are terrestrial proxies that are made by single or mixed sources. I'm going to spend a little less time on this than I might normally, considering the audience. But really, what happens for biologists, that's a big concern here, is that biologists get regolith simulants and they say, oh, I'm going to go use this, and that's a proxy for Mars. They haven't taken the time always to figure out in what way is it a proxy? Is it a mechanical property? What's the reason why this simulant was built, right? And so one of the things and we ran off just like everybody else to start and immediately started to recognize that we needed to take a more analytical approach and try and figure out what were these simulants for, right? There's also some issues with these Martian regolith simulants from a biological perspective, right? For one, the simulants are made on Earth, which means that the chances that there's no organic matter in there is probably pretty low. There's going to be organic matter in there, right? It also, in the case of Mars, none of these simulants have perchlorates, which I have to say, thank you for not selling perchlorate-laden regolith, because that would cause all sorts of complications for us. They might not be that accurate, right? So we have to work with regolith developers to understand, why was this regolith made, for what purpose, right? And how well can we rely on it as a proxy for biology, right? Also, Mars is a planet, right? We can make a simulant for Mars, right? Which is one spot. If we go 20 miles in a different direction, Mars is different, right? Just like it is here on Earth, right? The other question is, well, just like there's organic matter in there, how much of that organic matter is alive and how much does that affect our results? So actually, we have a little project that we've been doing in conjunction with Laura Fackrell, who's a friend of ours at JPL, and we're actually trying to investigate sort of the background of regolith simulants, what's in them, and how much of a concern, how much does that impact our biology? So we're doing this part where we're looking at how much organic carbon is in there. So we basically weigh it, burn it, and throw it in the furnace, burn off the organic matter, and then we figure out how much of that weight was, organic carbon. So if you look at soil, percent organic carbon in soil runs around 12% from the samples that we have. You look at, this is Mars Global, JES1, Lunar Highland Simulant. All three of these are from Exolith Labs here at UCF. Mars Mojave, which is a cheaper simulant that we can buy in bulk. We use it sometimes for really, really large studies. In one of the sites we'll show you, we did some work growing tomatoes for catch-up with Heinz. And we had to use much larger groups, amounts of regolith. And then JSC Mars 1A, which was sort of the first Mars simulant. You can see, for the most part, all of these, the organic content's actually pretty low. So there's not a high amount of organic content. At this level, we would not call this nutrient-rich material, right? So we're not going to grow a lot. The plants are not going to get a lot of benefit from this material. If we do, yeah.
So when you're doing, first of all, in the left chart, is that post-burn or pre-burn?
That's determined post-burn.
Right. So you have pre-burn measurements.
Yeah.
There, and do you ever remeasure like days or weeks later and see if it ever picked up anything from the air after that? Like see if the air changes those reagents. So.
Oh, so after we burn it? Yeah, I mean, yeah, if you leave it out, it'll collect stuff again. Yeah, so things will start growing in it.
So do you ever re-burn after that?
We haven't. No, so we just tried to get an idea of the baseline when we start. Just get some idea. We could try that though for sure. So here, basically, we just take some simulants and we put them in some peptone water, which is kind of a mixture used for extracting microbes and off the surface of materials like soils. And then we streak some plates and we see growth. So we dilute those out. We do some nice microbiology assays. This is done on what's called triptych soy agar. So with any of you have any micro experience, it tends to be a more generic kind of growth media. And what we see is that all the simulants definitely have stuff living in them, right? So we express this in what we call colony forming units per gram. It's kind of a backhanded way of measuring how many microbes there are per gram. So it's pretty high, actually. What I will tell you is that when we autoclave them, as long as the containers are the right size, so we use beakers about this big, we can put about 500 grams or 500 mils, sorry, of regolith in there, autoclave it and test it, it's all dead. So we can get that stuff pretty sterile pretty easily. We're actually doing some experiments right now to see how big of a container is too big. So we did autoclave a container about that big filled with used Mars Mojave, and the stuff in the middle won't get sterile. So there's still a bunch of microbes living in there. This is just when you use a normal steam-based autoclave system. So from that, we're kind of concluding we don't think that the organic content is a serious problem. We also don't think the microbial content is a particularly big problem either, as long as you sterilize in advance. We do have a little side project in the lab right now, which is actually isolating the microbes that we pulled off. And we have some different project I'm not going to talk about today, where we're looking at plant growth promoting bacteria that have been pulled off of the space station. And so we're running it through those assays to see if there's any good bacteria in the regolith already that might supplement, but we don't really see any evidence of that. So we think that this is a good sign that in terms of using the simulants as proxies, at least for a biological perspective, this part's okay, right? There's not a lot of early contamination that's making it problematic for us. All right. So what are we going to grow in this? So the first stuff I'm going to talk about is what in my lab we call the phase 1 stuff. In NASA, they'll often refer to this as pick and eat. So this is stuff that's not going to require any processing for you to get access to. So lettuce, sprouts, basil, right? One of the advantages of these is continual harvesting. You can go through and clip off some leaves, go make your salad, come back the next day, make some more salad, it's fine, right? A lot of times these have very short growth periods, right? Within less than a month, you're harvesting, especially in case of sprouts. All right, so here's some early work we did on this. We didn't have any other simulants other than JSC1A at the time. This was it. And we took some JSC1A, we put it in here, and we grew some lettuce. And with no supplement, after 30 days, we had some very, very sad lettuce. Actually, this is less than 30 days. Conversely, we add some nutrient solution, they did okay. It's very promising, right? So we thought, all right, maybe this isn't going to be so bad. So then we started asking about, well, what is it that if we could pull out nutrients that we need to provide to the plants, right? One by one, right? What do we really need to provide the plants? What's the little amount of nutrients that we need to provide in order to get the plants to grow? What's the essential element, right? Now, one of the things that all the regolith simulants are low in, which makes them really good mimics of Mars, is nitrogen, right? I literally will tell you that in my mind, the largest problem that plants are really going to face when we go there is the lack of nitrogen. There's simply not enough nitrogen for plants when they need what they need for growth. It is a rate limiting component. So you guys ever seen fertilizer and it says like 15, 15, 10 or 15, 30, 15? That first number is the nitrogen content, right? In that fertilizer. It is a rate limiting component in plant growth. You need a lot of nitrogen for plants to get large biomass. So here we did a study comparing JSC1A and Mars Global. So now we've moved forward a few years and we're looking at if we get a full supplement of that Hoagland's nutrients, which is kind of a hydroponics nutrient media that we use a lot. This is Hoagland's is where the starting point was for some of the veggie work that they did in the space station. They've kind of moved to a different nutrient media now, but this works for us. And in 1X, after 30 days, we get good growth in both simulants. If we deprive them of nitrogen, phosphorus, and some potassium, the plants are dead. So there's not enough in the simulant for them to acquire that material. If we remove the nitrogen, they're still dead. If we just take out the phosphorus and the potassium, actually the JSC works pretty well. but MGS still really suffers. And what we've learned now, and I have a master's student who's kind of doing some work now that's looking at the differences in washes of the two different simulants. JSC is made of material that has higher solubility than Mars Global. So it turns out Mars Global, with its improved mineralogical accuracy to the surface of Mars, makes it harder to grow plants in, but it's a more accurate simulant for what the surface is, right? So this is interesting on our side of the community with working with regolith simulants, because a lot of the very early work with JSC 1A was like, oh man, plants are going to go great in this. It's going to be great. Yes, and that was important literature to establish the field. But it turns out that as you move to more mineralogically accurate simulants, the problem becomes more difficult because it's not as water soluble, right? So our conclusion for this is that JSC 1A has more soluble components than Mars Global. Like I said, I have a master's student who's doing much more extensive work on this. But it's interesting that we had that data a couple of years ago, and it wasn't until we started specifically looking at that question where we really started to understand what this data meant. And so it's an important consideration because you still have a lot of biologists that are picking simulants because plants grow in them. They say, well, I can't get my study to work in Mars Global, so I moved to Mars Mojave. And I'm like, but Mars Mojave is not a good model, so don't you see the problem? And they're like, yeah, but I can't do the study if I don't do it in Mars Mojave. I'm like, okay, well, all right. So how are we going to introduce nitrogen? So this one, I'm just going to show you a little bit of this. This is some work that's actually multi-generational in my lab. So it started with Cynthia Montanez a few years ago, and then Leah, Haley, and Indigo have kind of followed up on this project. Haley's really, she's sort of I think the queen of A. cylindrica in our lab right now. So Anabaena cylindrica is this filamentous cyanobacteria that's really cool because it's capable of both doing photosynthesis and nitrogen fixation. So it can take nitrogen out of the air and fix it into biologically usable forms. That's our fertilizer, right? And so these are images she collected of cultures that she's grown in the lab. This is a scanning electron microscope image. It's actually this fiber twist you see here. That is a fiber of A. cylindrica on the surface of regolith. So she can capture an image of that. Leah built these bioreactors in her first semester in the lab off of some 3D printed parts and just sort of some idea of how to use regolith as a substrate for growing this material, right? So, and it turns out that A. cylindrica actually grows pretty well in regolith. And so this is one of these great stories that is kind of born out of the pandemic. So we had these cultures that were inoculated of regolith with A. cylindrica. And then my students all got kicked out of the lab because of COVID. And I basically would come into the lab and I had to make the rounds of watering everything and keeping everything alive, streaking plates, doing culture work, stuff that professors are not good at. And I kept things going, but mostly I forgot to take care of some stuff. And after 6 months, we literally had a hockey puck of regolith. This A. cylindrica had formed so much biofilm over it that it become like a dense mass, all grown over with A. cylindrica. So here, this is just showing you, this is different types of A. cylindrica cultures kind of on regolith as well, right? So we're looking at different regolith particles, and you can see the material, these microbes that are in association with it. They're also changing the structural composition of it, right? So this is regolith under the electron microscope, kind of jagged, right? As we introduce biology to it, it becomes softer. The technical term in the lab is fluffy. So it becomes more soil-like, right? It becomes fluffier, it aggregates material better, it holds water better. It starts to act like soil does. But can it help us improve growth? So this is a preliminary project that Haley was working on. She's actually trying to get this up for her senior project. So here up top, we have Arabidopsis thaliana plants, sort of our E. coli of the plant world, growing on soil. Down here is Arabidopsis thaliana growing on straight regolith. So not so good. Here we have Arabidopsis growing on regolith in which A. cylindrica grew for 4 months. So we're refining this process. We're trying to get ways where we can quantify the amount of acylindrica that grew in there. But inherently, it's telling us that we're actually improving the nutrient content of this material. We're starting to look into how much nitrogen we're actually adding, how much carbon we're adding. So, you know, we're getting into a mode where we're starting to see this as a possible way of providing nutrients into the regolith and beginning to convert it into soil. All right. So that's, you know, kind of a summary of where we were looking at phase one, right? We're trying to take regolith simulants, we're examining them, we're trying to identify ones that are more compatible with the biological testing, and we're trying to build a pipeline where we screen for, you know, initial contamination in the regolith simulants that we get. So what kind of biology is going on in there, what kind of organic content is going on in there. This is now going to be the way we run things in the lab, is that when we get simulants, we are going to go do these, and this is going to be part of our initial battery of testing. What microbes are in there, what's the microbial load, what's the organic content. That's all going to be part of what we do to pre-check our regolith before we go do our biology. We're trying to get everybody on board that this is just part of what your best practice should be. If you're going to do biology and regolith, it pays to know what's in it already, right? So then we're looking at these different methods of converting regolith into soil-like material, right? But then we started to contemplate phase two. What the phase two plants are things that are not pick and eat. They are things that require longer periods of growth. Things like bushes, strawberries, tomatoes, right? Cucumbers, plants that are going to require a more extensive root system. They require greater amounts of nutrient input. There's actually sometimes not as simple as like, oh, I plant my plant and it makes flowers and then it makes fruit, right? You actually have to optimize the nutrient content of the material and the growing time, temperature conditions to optimize flowering as well as fruiting. Some of these even may require pollination, right? So this was sort of the next step up, right? Was adding some increased complexity, but this is where a lot of that varied diet is going to come from, right? Even peppers, right? There's a big pepper challenge about who can make hot peppers for astronauts. Well, if you grow those peppers on Mars, you're going to have to contend with all of these additional issues. So we actually had a weird opportunity to do this when I got an email that I thought was most certainly spam from Heinz that said that they wanted to talk about growing tomatoes with me. And I went, of course you do. And I totally ignored it because I said, all right, I'm not going to click on that. IT tells me I shouldn't click on emails from strange people. And so I got another email from them and I went, okay, you guys are really insistent, but I'm not doing that. And then I got a third email and I went, okay, what else am I going to do? So I read the whole thing and I went, okay, I think this is legit. So I sent it to our Office of Sponsored Programs. They reviewed it and they went, this is real. You should call them. So we had a meeting and they said, well, you know, we want to try something crazy, right? This is a time when we want to try something to build morale and do something silly. And let's go see, can you grow tomatoes in your Martian simulants? And I went, I don't think so. I think that's really, really hard to do. And they're like, well, we'll give you guys some money to try it. And I was like, well, okay, then we'll try it. Let's do that. So we decided that we knew enough, at least to give it a shot. And so we started this project to make the Mars edition catch up with Heinz. Now, this, for me, the intellectual curiosity here was an opportunity to bring lab scientists who work with plants and agricultural scientists who work with plants together. Lab scientists think about plants by saying, I'm going to give you just these nutrients, and then I'm going to sit and watch and see what you do. They're hands off, right? Agricultural scientists go, are you dumb? That will never work. You need to do this to the plant. You need to wait for 4 weeks. Then you change the nutrient condition on them. Then you change temperature. Then you got to give them this much water. Then you have to withhold water. So they had a whole different regime for how you actually produce food, right? As opposed to what most of us do in biology, which is to watch what the organism does, right? We're trying to understand, when I put the plants in regolith, how are they going to behave? And they said, well, you need to tell it what you want it to do. You need to give it the nutrients and program it, right? Program the biology to behave the way you need it to. So that was what really interested us, an opportunity to talk to agricultural scientists and kind of play with those nutrient profiles. So we bought a giant tent and inflated it inside of a building so we could have environmental controls, which is was interesting because the facilities people thought we were crazy. The people who made the tent also thought we were crazy because we asked about the height. And then I said, no, what's the height on the outside of the tent? I'm like, why do you need the height of the outside of the tent? Well, I need to know if it's going to clear the roof of the ceiling. And they were like, are you inflating this indoors? And I'm like, I can't really talk about that. But yes. So we inflated this structure and built this room in here where we could essentially have a reasonable amount of climate control. This space allowed us to grow enough plants to make about 1,000 tomatoes. In this case, unfortunately, we had to switch to Mars Mojave because they're simply not cost effective to use Mars Global or some of the other simulants. So we used Mars Mojave for this, accepting that that was not going to give us the best results, but it was going to give us something to work with so we could say whether or not this is even feasible. The neat thing about this, none of these seeds, we got the seeds from Heinz, we took them into the room. The plants that grew in this project never saw the light of day. In fact, they saw only LED light, and we now have 3 generations of plants in that group that have never seen anything but artificial lighting. So we did some early germination, early growth studies. So they gave us 4 varietals to work with. And it's interesting because they talk about these varietals like they were pets. When we were talking about how they grew, they said, well, of course, yeah, that one's going to do great in there. It always would. Yeah, we knew 1428 would do great. And they're like, that one's going to be your winner, I bet. You know, like they actually had discussions with each other about it. And they know things about these different plants, where they grow, what places that they find them in, and where they use them. So these are all lines of tomatoes that Heinz actually uses to make ketchup. So, which is kind of cool. So we did an early stage growth like this, and we got into this stage, our mid stage here, and then we transferred them into these big pots, which were very, very difficult to fill. So there is another COVID story here, and I have to stop and praise my students because we had the tent set up, we had all the plants here, and they were about ready to transfer, and we got the email from the university, we're closing. Your students have to go. You have what you can get. No grad students are allowed. Whatever science is going on, you're going to have to keep running it for at least the next 2 weeks. And I went, we needed to start this. Like this needs to start. Like we're going to lose all of these plants if we don't migrate them over. So I didn't know what I was going to do. I left the room, I left the tent, and I had about 6 or 7 students who were all working in there, filling containers and everything else. At about 2 a.m., I got an email that said, we're done. And it was a picture. They had stayed and set all of these plants up in all the pots. They filled every container, they got every plant transferred, and then they basically gave me instructions on how not to mess it up. They're like, do this and it'll be fine. I'm like, okay, I can do that. So basically for the next 2 weeks, I made my rounds. I drove to the lab and I streaked all the plates and kept all that stuff alive. And I drove over to the space where we had the tent and I sprayed all the plants and kept them going too. And then we were allowed to have grad students back, and so they helped it back over. But I really can't say enough about how dedicated the students were to this project and how much it meant to me to have them do that. All right. So we wait a couple months or so and we get to the mid stage. We have a lot of plants. It's not great in this picture, but you can see there's a lot of what we call frizzy leaves, curled leaves, the signs of stress. The plants, they don't like this environment. They're growing in it, but they're okay, right? So the question is it the lighting? Is it the water? Is it the regolith? Yes, that's the answer. It's yes. It's, you know, all of it is complicating factors. And so we've been working now, we have a new room actually. So we've ditched the tent. We have an entire room now that we're doing using over at Florida Tech, where we're trying to basically create a more climate controlled chamber. The lights are further up. We have more control over the amount of lighting. So we're playing in with that factor now. So did we get plants? Yes, we actually got fruit too. So this is 2401, one of the varietals. You take a look. This is in soil. This is our soil control in our greenhouse. This is from the Mars Mojave simulant. So they are significantly smaller. When I showed them pictures, they were not happy. They were like, this is kind of rough. And so finally we had a long conversation and I felt really bad. I was like, guys, I'm really sorry. And then finally one of them was like, has anybody ever gotten tomatoes to grow in this stuff before? And I'm like, no. And they're like, well, this is a win. I'm like, thank you. Yes. I was like, this is a win. I was like, we did something cool. Right? So that was promising. When we open them up, we can see, this is what they look like. And they're smaller. They're a little more acidic than regular tomatoes. I put I didn't have it on here. So there's a test that you do for tomatoes called a Brix test. It's sugar content. And that's really important for ketchup making, because I didn't know if you knew this, but ketchup has a lot of sugar in it. And so it's one of the things that they're very interested in is Brix content. Our fruit has less sugar. It's more acidic, less sugar. That has to do with nutrient delivery during flowering. So as they flower and they convert to fruit and they start fruiting, if you miss certain nutrients aren't getting fed in properly, like the balance of potassium and calcium, that can influence things like pH content, sugar content. And so there's something about how they're taking up nutrients while they're in these simulants that is skewed in a way. We're starting to think about how we could add other nutrient supplementation and sort of change the pattern. What we don't necessarily want to get into, though, is a system where someone growing food on a settlement on Mars has to come up with a whole different nutrient plan for every type of plant that they're growing, right? That's a lot of extra work. So we've got to come up with some uniform systems, right? But this is telling us that it is possible to do long-term growth in here, right? So if we look at the total number of fruit per plant that we were able to pull out, right? So here is Mars Mojave simulant, and here's soil. So yeah, soil beats us every time, but we actually get a reasonably decent amount of fruit, right? So by Heinz's standards, growing on farms in rich, nutrient dense soil with professional farmers, they didn't think much of our results. But for growing in challenging simulants under difficult conditions that would simulate a artificial lit environment, this was a pretty good success. So we're pretty proud of it.
Are the error bars and on the plots, are those like the various, just the ranges you got from the different
It's just the ranges from the plants, yeah. So the letters are telling us anything with the same letter are statistically identical, right? Or they're not statistically distinguishable, sorry. And things that are different letters, that means they're statistically different. Right?
base level? It's almost the same. right?
Yeah. I mean, it's not too bad off, right? So we were pretty happy. We went and did a second round of this and we learned a little bit of improvement. That data we're actually crunching now. We're getting some metabolomics data on that now. So we actually farmed that fruit out from that study out, and it's being analyzed now to really dig into the metabolic pathways and how those are changing. That's sort of a new area that the lab is moving into, is we started bringing a lot more rigorous analysis into the things that we're growing in space biology, because that's actually kind of missing from the discipline. There's not a lot of the transcriptomics, metabolomics, the omics suite that biology lends towards everything on Earth. We need to start doing the same thing, because those can give us real clues on where the stress factors are, right? What's causing the stress in the plants and how to mitigate that, right? So we can use that to our knowledge. The seeds are viable. We've taken the seeds from this. They're about the same viability as the seeds that we grew in soil. So there's no loss of viability. We've actually done 3 generations now in regolith, and they're all fine. So.
Did you taste them?
I did.
And were they?
They are acidic and not as sweet. So we sat and did a side by side where we cut the tomatoes and we taste tested them, and we're like, yep, they're edible. They're totally edible. They're not bad. But when you eat one tomato, that's like, you can taste that you can very much taste the difference.
Have you noticed any improvements across the generations?
A little bit, but we're not clear how much of that is also improvement in our green thumbing, our red thumb, so how much better we are getting at working with the regolith. So that's something we have to be a little bit cautious about in multi-generational studies. But yes, there does seem to be some improvement. We're actually talking now about trying to do some epigenetics. So looking if there's any switches that are going on at the genetic level that might be changing as well. So. All right. So with the last 3 or 4 minutes I got, I'm gonna try and tell you a story about some perchlorates. So this is a project that I was very, very happy to get the chance to participate in. So on the surface of Mars, perchlorate concentrations can be as high as up to 2% weight by volume. That is higher than anywhere you will find on Earth. The places on Mars with some of the lowest concentrations of perchlorates are still higher than most of the places that you will find on Earth, except for the Atacama Desert in Chile, which has got the highest concentrations naturally occurring, right? They're made through a variety of processes. I won't get much into, but one of the things that we know is that 1% perchlorate. So we went and did a test in our lab. We made some perchlorate laden Martian regolith simulants. We did both JSC and we've done Mars Global. Loaded them up with some perchlorates, which was very challenging to make with all the safety procedures. What we found is that 1% weight by volume is lethal to lettuce, radish, peppers, tomatoes, tobacco, arabidopsis, clover, cucumber, watermelon, basil, and cilantro germination. So pretty much lethal, right? We take plants that we've grown outside of it, we plant them in our perchlorate regolith, and they die. So perchlorate also can bioaccumulate. You can bioaccumulate, so if something actually survives being in the perchlorate, it will pick up the perchlorate, and then when you or a Martian settler eats that, they will get cancer from that. So there's a really big issue that has been largely sort of black boxed, and a lot of settlement research for Mars is, well, and we'll take care of perchlorates. And I was like, well, what are you going to do inside of there? Because any changes you make are likely to change the mineralogy of the simulant. So what's it going to look like, right? And so I was very fortunate. Anka reached out to me from Arizona State and said, hey, I read your thing about this. I know how to fix this problem. And I'm like, okay. And she's like, I'm a microbiologist and I have bacteria that eat perchlorates. All right. So she's now working on a project where we got an NSF project where she is going to use her microbial community to pull the perchlorates out. She's using the perchlorates essentially to drive a carbon cycle to bring organic matter into the regolith. They're going to send it to us, and we're going to be testing plants in it, which is pretty cool. Yeah, this is that. All right, and then the last thing I'll say very, very, very momentarily is one of our projects that, what are we going to do with all that inedible biomass? So this is a work that started as a Plant the Moon challenge, and the team lead on that, Katie Hubert, basically took that into the lab because I was like, you're kind of reaching the limits of this competition. You need, and she was like, yeah, I feel frustrated because there's all these questions I want to answer. And I was like, okay, let's bring it into the lab. So now it's a research project in the group. This is what she's doing her senior thesis project on, is basically using inedible biomass to grow oyster mushrooms. So here she's got a really nice picture of the mycelium of the mushrooms actually coating regolith. And she's got several rounds of data now showing that, again, the introduction of these edible decomposers like mushrooms essentially take this blend of regolith and inedible biomass and begins to convert it into soil. But we also get mushrooms, which I like mushrooms a lot. That's pretty great. All right. So in conclusion, I hope I made a compelling argument that while we've got a lot of ground to cover, no pun intended, for regolith-based agriculture, that it is a necessary complement to hydroponic models for how we're going to survive on, for certain, on a Martian settlement. I think it would also be really useful on a lunar settlement. So I think, again, anytime you increase the routes to food production, you're increasing the resiliency and food safety of that system, and that's mission critical, right? I think we need to be stressing more as biologists what your simulant is for, and thinking about how you handle the simulant just like you handle your media, right? So when microbiologists make media for growing bacteria, you don't whip it up and pour plates, you go sterilize it first, right? You run some tests, right? And so we need to take the same practice to this media and this substrate. That we need to be thinking about all of the aspects of regolith, including the stuff that seems challenging for us to grow plants in. We need to be working on this from both ends. We can't ignore the perchlorate problem. We can't wait until the perchlorate problem is solved, right? So we have to be working in both directions. And I think, even though as we move away from JSC1A, we see that the work is harder, it's more challenging, we're developing increasingly better models of how we can actually convert these simulants and hopefully Martian regolith into soil-like substrates where we can grow plants effectively. And maybe along the way, bring along some of the microbial companions that have kind of helped us do all of this on Earth. So with that, thank you for your time. I'm happy to take any other questions you guys have.
So in the picture with the plates, do you know which bacteria are more present in them? Like which ones are the highest in abundance, like species wise?
Oh, these?
Yeah.
We have not done any identification yet. I will tell you that that is part of the project. So, Laura's actually identifying what's actually in there as her end of the project.
Did you publish your Anabina paper? Or
Not yet.
Okay.
We're working on it. So, there's a lot more that we're trying to get out of it. At the time, this was sort of a preliminary run through. We're working on that right now, actually.
About that paper, actually, did you look at the metal content of the plants grown in it too, or just like?
We haven't yet, but that's on the docket. So we have two different routes where we're looking at metal accumulation. One route is we're starting to look at whether we're getting significantly higher spikes by like EDAX. And then we're also starting to do ion chromatography and looking at the metal content that way.
Okay. More question, go ahead.
I was just wondering what plant growing bacteria that was swapped on the ISS. Do you know what?
Oh, so actually what we get from the space station is they swab down the plants on the veggie system.
Okay.
And then once they get back to Earth, they basically isolate them out and they identify them. And we basically get shipments of microbes of different species that we test out. So I think we've tested about 20 or 25 right now that we are screening for plant growth promoting properties. So we have like 4 or 5 assays that we use for different common plant growth promoting phenotypes, whether or not they can help solubilize phosphate out of the environment, altering plant hormone production, a few other things. And so we put them through these screens, which are plate-based assays. So we can just look for, color changes, halos on plates and stuff, collect that data, and then we kind of mark that out. And then we go and before we do any of that, we get a list of the species. We go to the literature and determine whether or not any of these have been previously identified as plant growth promoting microorganisms. We immediately star those and test those out first. And then we test out the other ones to see, one, have there been any changes maybe that haven't been identified before? So we're trying to pull from that and get that information. So we published one paper, the first round of that, we published 2 years ago in Frontiers. We're actually starting to compile now sort of the next generation of that, which has got a couple of other assays. We're also now beginning to incorporate the microbes into random positioning microgravity simulators. And we're starting to do plant and microbe together. And, it's artificial, right? Because in an RPM, you're not actually creating real microgravity. It's just, it's just spinning it, which is meant to confuse the plant. But there are significant changes in gene expression in those systems. And one of the things that we think is that might affect symbiosis, so how well they're able to integrate with the plant. One of the other big differences about our microgravity simulator is we've seen some of the other microgravity simulators, and the light source sits on top here, and then the plant rotates. So the problem with that is that the light source stays from the top, which means you actually have two different confusing variables, right? Because as you turn this way, the gravity machinery in the plant gets confused, but it's also confused because light's still up, right? So the position of light never changes, but it's different because the shoot port of the plant is now facing down. So you're actually giving it two different inputs. And we think that actually overrides because the plant can still sense that way's up because there's light. So you're actually getting imprecise effect. You're not necessarily teasing out gravity the way you'd like to. So we worked with some engineers at Florida Tech, and we built a simulator where the lights are in line with the plant. So no matter what direction it turns, the plant is getting light on top.
So you have that piece of equipment at FSI?
Yeah.
Is that in your lab?
Yeah, we had some engineering senior design project. They built it as a simulator. Yeah. So
It's nice. Thank you.
yeah. Well, we're working on hopefully getting some other ones built. So it was a lot of fun.
Okay, great questions. Yeah, go ahead.
For the process you're talking about getting the microbes on the ISS, is radiation a concern? Anything die off?
Well, so we get whatever comes back down, right? So if it survives, right, then the argument is if it's the whole premise of this is that if it survives, then it has spaceflight history, which makes it easier to make the argument for putting it back up there. So because what we think is that I mean, I think even if you talk to a lot of NASA people say the idea of keeping these environments sterile doesn't really work, right? So they've published tons of papers now on the microbiome of the space station because you can't sterilize human beings, right? So you send us up into space, all the microbes on us come right back out and populate the space station. So it's not going to work. So these microbes that have spaceflight history, they survive that process and we can send them back up. One question we have is whether or not things that were previously identified as not plant growth promoting could, being in space, create mutations that might facilitate that, or do they become pathogenic? So the end of our pipeline ends up with a test where we grow them in conjunction with plants. What we're going to add next is we're actually adding a test with nematodes, where the nematodes are going to eat the microbes and see whether the microbes are toxic to them.
Okay, great. Please let us know if you have any questions from online participants. You can unmute yourself. And Lance, go ahead.
I want to know if you did like XRF on the tomatoes or like on the any testing on the plants themselves instead of the soil and substrate.
Right. So like I said, we've got this most recent batch. One of the most recent batches of tomatoes is actually out from mass spec. So we're going to be doing the metabolomics on that. So we'll be able to see like secondary metabolites. And if you're asking, have we done like metal content analysis? We did a little bit of that. So one of the concerns that we had was chromium accumulation, especially from Mars simulants. So there's a we didn't think of it in terms of that for chromium content, but
MMS.
yeah.
Okay.
So well, it's there normally, but the challenge is we hadn't thought about it because we hadn't thought about the bioaccumulation when you make a product. So what Heinz thought about was where you're going to take all those tomatoes and you're going to blend them, cook them, and concentrate them. So that metal content is going to get higher. And then we were like, well, I mean, but it's relatively low still. And they were like, well, you're not thinking about our target audience. And I was like, what do you mean? And they were like, who eats ketchup? And I was like, oh gosh, kids. And they're like, yeah. So like what's not a harmful dose accumulation in adults could be very different for children. And so there was that was something we talked about a lot, was whether or not that needed to be factored into this. And that became a new element that we're starting to figure out how to work that into our pipeline is, when we get these plants, we get the product coming off of the product line. How do we analyze them and what are the factors that we need to consider, right? Because it seems like in a lot of space agriculture, whether or not you would eat the food has not actually been considered very often, right? So that's like asking about, does the tomato taste good? It's like, what does that have to do with anything? Like it's kind of funny. It's like an afterthought, but I'm like, so actually one of the projects I have right now, when my student's Ty, I've got him on this kind of absurd idea for something we're calling the yum yum factor, which is there a way you could pull together elements of mass spec like metabolomics and some other things? Like, could you pull together a data set that would say this would probably taste good, right? So like you could be screening your system, and from all the data we're already collecting, could you put that together and say, oh, and it might also be good, which would be nice. So.
Would you take something that does taste good and analyze that or something like that, and then
Yeah. So like he's looking at he's been looking at a couple of different things to do that analysis with, and so he's trying to pick a couple of metabolites. It's I mean I do feel a little sorry to test him.
It's a big project.
It's an interesting project, right? I mean he's gonna be starting his PhD in the spring, so he's got a while. Gotta hope he's not on the call.
Okay, I think let's thank our speaker again. And I will end the seminar now, but we can continue this discussion.
Yeah.